My name is Venera Tyukmaeva, and I'm a lecturer at the University of Liverpool. My research focus is host-parasite interactions, and I study trypanosomatids parasites in Drosophila. Trypanosomatids are quite diverse group of parasites, and they infect correspondingly diverse array of animals and plants, even though they are most famous for infecting humans and causing dangerous diseases. Relatively recently, trypanosomatids have been discovered as common insect parasites in the wild and in different species of Drosophila as well. While trypanosomatids of medical importance change their host from mammal to insect, in majority of insects, they just stay within one species and genus. And that's the case for Drosophila, where the parasite has much more intimate and more evolutionary ancient relationship with its host. For all host-parasite interactions, there are always two sides. There is host and there is parasite. And I worked with NEOF to understand the genetic basis of parasite side of this relationship. For the first part of the project, I sequenced a high-quality genome. And this is very important for me because it will allow me to understand how the parasite is related to other trypanosomatids. Also, I sequenced transcriptome, and it will help me to understand how parasite tricks host immunity to establish inside the host. I had a really great experience working with NEOF. Especially, I would like to highlight their professional support team. They worked me right from the start, and they helped me with the advice which technology to use. They helped me to design my project for the best outcome and the lowest cost. They were checking my samples, and they consulted me on the results. Also, they offer training, which is very useful for people who start with new methods and new technologies. The NEOF project is fundamental for my future research. The information which it gave me on genome and transcriptome is very important to understand how the parasite interacts with its host. But it's just the beginning. It's a first step for me to understand how the trypanosomatid parasite interact with insects. At the moment, I'm working on the publication on this project and writing a future grant proposal.